a groundbreaking discovery is shining a new light, literally, on viruses. Researchers have found a way to listen to the unique vibrations of these tiny invaders, unlocking new possibilities for biological imaging and antiviral research. Leading the charge is Elad Harel, an associate professor at MSU, who has dedicated his career to exploring the unseen world of molecular motion. Specializing in ultrafast spectroscopy, he uses short laser pulses to capture the dynamic dance of molecules, revealing how microscopic forces shape complex systems. One of his latest breakthroughs? A revolutionary microscopy technique that doesn't just take static snapshots but allows scientists to observe biological landscapes in motion. This pioneering work has earned Harel the prestigious MSU Innovation of the Year award and the university's first-ever grant from the Keck Foundation. Now, in a study published in the Proceedings of the National Academy of Sciences, Harel and his team have achieved something remarkable. They've used light to listen to the sound of a virus, capturing its nanoscale vibrations in real time, a discovery that could change the way we understand biology. But Harel didn't do it alone. He collaborated with Do-in Pian, a professor in MSU's Department of Microbiology, Genetics, and Immunology. Together, their teams worked to provide virus samples for this ambitious experiment. Teamwork really matters in a project like this, said Yaqing Zhang, a postdoctoral researcher in Harel's lab and the study's first author. It's fascinating to see these tiny virus particles breathe under laser illumination. For Zhang, the implications are enormous. This technique can be applied to millions of viruses and biological samples, he explained. The more we understand them, the better prepared we'll be for the next pandemic. Hearing the unheard, every object in the universe vibrates at a natural frequency, whether it's a distant star or a virus. Harel describes it as a kind of molecular music, where atoms move together like tiny balls connected by invisible springs. The idea of listening to viruses was uncharted territory. Scientists had previously studied ultrasonic vibrations in metal nanoparticles, but no one had asked whether biological systems could produce sound when exposed to force. Harel's team designed an experiment to find out. They used short pulses of light to set the virus into motion, then a second pulse to capture that motion at different points in time. By stitching these moments together, they created something never seen before, a molecular movie of a virus in motion. What they discovered was astonishing. Viruses do, in fact, have a unique vibrational signature, a sound all their own. This revelation paves the way for a completely new approach to studying biology, one that could identify viruses, proteins, bacteria, and even cells based on their natural voices. A new way to see the invisible. The breakthrough addresses a fundamental challenge in biology. How can we observe living systems at high resolution without disrupting them? Electron microscopy, one of the most powerful tools available, can capture incredibly detailed images. But there's a catch: it requires samples to be frozen or placed in a vacuum, conditions that are far from the real environment of a living organism. With their new method, Harel and his team can track biological motion in real time, under natural conditions. They spent years refining their ability to measure acoustic vibrations at the single particle level, working closely with Pian's lab to study various viruses. One of the most exciting aspects of this approach? It eliminates the need for complex labeling. Traditional imaging techniques often rely on attaching fluorescent markers to molecules, a process that can be slow and invasive. But Harel's method allows researchers to detect a virus using nothing but the sound of its own materials. The sound of a virus breaking apart. So what do viruses actually sound like? The answer is hidden in their vibrations. Harel's study found that viruses oscillate at frequencies in the gigahertz range, far lower than the optical transitions used in conventional spectroscopy. Even more intriguing, they observed that as a virus starts to rupture, its acoustics change. Like a deflating balloon, the sound shifts to a lower frequency, revealing the process of viral disintegration in real time. The future of biosonic spectroscopy. The next step is even more ambitious. Harel's team wants to track a virus as it moves, mapping its journey from the moment it enters a cell to its final stages. Current methods, like electron microscopy or fluorescence imaging, make this process slow and challenging. But with biosonic spectroscopy, researchers could watch the full life cycle of a virus unfold, offering new insights into how antiviral drugs and treatments work at the molecular level. The U.S. Defense Threat Reduction Agency has already taken notice. They're supporting this research in hopes that it could accelerate the development of antiviral drugs, potentially revolutionizing the way we fight infectious diseases. With each new discovery, Harel and his team are proving that sound is more than just something we hear. It's a powerful tool for seeing the unseen, a key to unlocking the deepest mysteries of life itself. Let me know if you'd like any tweaks or further refinements. Thanks for watching. If you are new to our channel, please subscribe to our channel and turn on the bell for further notification.